So my job today is to talk to you a little bit about how we're using the new findings that are coming out of genetics research and brain imaging science to start thinking about how we will develop the new generation of treatments for kids and youth and adults with ASD. So this has been said already that we all are very familiar with this saying that if you know one child with autism, you only know a child with autism. But I want to tell you that we have actually proven that this is true. So not only on the diagnosis side of things, where we know that in order to give a diagnosis of autism to somebody, they have to have social difficulties and repetitive behaviors, but in practice, people with autism across the lifespan will present with a variety of differences. Some of them are weaknesses, so things like irritability, aggression, anxiety, ADHD-like symptoms, sleep dysfunction, sensory issues, higher incidence of epilepsy, for example. Some of them are just variability. So when it comes to our cognitive skills, our thinking skills, we'll have some people who are functioning in the intellectual disability spectrum, where some other people with autism will have superior cognitive skills. And some of them have to do with actual absolute strength. So for example, a large number of people with ASD can process detail in the context of lots of noise information better than typically developing people. So you can imagine now that if you develop combinations of those circles, so combinations of those differences, you truly come up with very many different types of ASD. If you look on the genetic side, what happens is we're confirming that this amount of heterogeneity, this amount of variability, is actually in the biology to begin with. So we have more than 400 genes now that have been associated with autism, and we have explained about 20% of the autism variability that we see. So we expect this number to grow significantly. So now if you're somebody like me and you're trying to develop treatments for autism and you have 400 different autisms, you're in a little bit of trouble, right? So it turns out it looks like a little bit better than we originally thought when we got those big numbers, because what's happening is you can start grouping those types of differences into metabolic pathways and brain pathways that look similar. So instead of having 400 different types of autism, you may in fact have 15, 20 types of different biological forms of autism. So one way to do it is to group the genes in clusters that have the same effects on brain. But again, I told you before that only about 20 to 30% of kids will have a genetic mutation found. So how do you group kids who do not have a genetic mutation? And this is how the brain story becomes very important, and this is where we employ our friends, the mice. So there's a few million years of evolution between mice and humans, but actually quite a few parts and circuitries in the brain are conserved. And so what we are doing, what our collaborators are doing, is they're taking human mutations that cause autism, so human genetic differences that are associated with autism, put them in mice, and then we study their brains so that we learn what the effect of the mutation is on the brain. And once we do that, we cluster then all the different type of mice in groups of mice that have more similar brains, because we are assuming they have more similar biology. And then we take this to kids, and we're trying to figure out if we can learn from this clustering exercise in the mice in order to cluster the kids that we don't have genetic mutations. And I just lost my earpiece. So my last couple of comments is, so when we're thinking about developing new treatments for kids with ASD and youth and adults, one approach is to go after the targets that we learn from the genetic testing, because we can map them onto different brain pathways, and I just put some examples for you. Another way is to look at the brains of kids who have passed away from other causes. We're learning from their brains, and this is how the neuroinflammation story came about. So we were developing drugs and treatments that go after neuroinflammation. And my last thing is to mention that we can actually learn from systems neuroscience, where we're looking at what circuitries in the brain are involved in the areas of interest, and then we go and manipulate those circuitries. And my last comment would be that just to remember also what the purpose of treatment is when we are trying to do this. So we are trying to sift the biology in a way that makes learning easier, so that we decrease distress and dysfunction in kids and youth and adults with ASD. Thank you.